on. Now, what I'd like to talk about today, and I do apologize for showing this slide so soon after lunch. I did go to an International Society for Wildlife Disease meeting a little while ago and swore that I'd never go back to one of these, but it does make the very graphic point that emerging infectious diseases do represent a formidable challenge to the conservation of animal and plant species in the 21st century. There are many cases now where pathogens have had dramatic impacts on populations and species abundances, and these in turn have had impacts on community structure and implications, in fact, for ecosystem functioning. So, for example, in the recent decades, there have been losses of something like 30 amphibian species, and of those that remain, about 30% of those are seriously threatened. And that represents, in fact, one of the most spectacular losses of vertebrate diversity so far on record. And that's been due to a chytrid fungus, which has been devastating these populations. Similarly, in Hawaii, avian malaria has resulted in the loss of about 10 bird species since the 1900s. The Tasmanian devil, there's been a facial tumor that's been ravaging through this populations of this species and threatening its very existence. There have been losses of populations of turtles and sea urchins due to various viruses and, in fact, an unknown pathogen that you may hear a little bit more about later on. The fact that the die-off in the sea urchin populations in the Caribbean resulted in something like 93% mortality of the diadema and has, in fact, impacted on ecosystem functioning in the Caribbean. So we have lots of examples of diseases causing population declines and species extinctions, particularly in terrestrial systems. But for the moment, we know relatively little about the impact of diseases on marine populations. And this is despite the fact that there have been decades of study really on factors like biological and physical agents structuring natural communities and marine systems. In this particular study, Hervel and colleagues looked at the number of mass mortalities there have been since the early 1900s in the marine systems, and you can see here that there have been what appears to be an increase in the, in the number of outbreaks in recent decades. There have been mass mortalities in things like sponges and sea fans and corals, sea urchins, some invertebrates, as well as vertebrates like seals and also fish. But the question that arises is this really increasing prevalence, or are we basically looking at increased effort? So that's something that is particularly a question here on the Great Barrier Reef and leads us to some of the questions that we've been posing in our program within the Center of Excellence. How healthy is the Great Barrier Reef? There certainly is a tacit assumption out there that the Great Barrier Reef is healthy, that corals don't get sick, but corals are animals, and just like any other animal and plant species, they are subject to pathogens and disease. In fact, corals are more than just animals, and you've been hearing a little bit about that this morning. The coral is an animal which associates with a unicellular plant, the zooxanthellae, and you've seen the graphic impacts when they lose them, when that association breaks down and the corals turn white and bleach. But even more than that, the corals are associated with bacterial populations that live on the surface of the coral tissue. So this is the surface microlayer, or the mucus, sitting on the surface of the coral polyp. And it appears that the corals are actually associated with dozens of different types of bacteria in that layer. And in fact, recent research is raising the possibility that, in fact, this mucus layer may actually function as an external immune system. So if this is the surface of the coral, and here's the water mass, the sea here, between the coral surface and the sea, you have this layer of mucus. And within the mucus, there are, as I said, dozens of bacteria. And recent research is suggesting that There are some of these bacterial species that have antibiotic activity, and in fact may be beneficial to the overall health of the coral itself, that some of these are able to affect up to 10% of the functioning of other bacteria within the mucus. And even more interesting, if there is a change in these microbial communities in the mucus layer, then it appears that the corals may be more vulnerable to disease. And in particular, this study showed that when there was a bleaching event, the change in the microbial community was such that there was an increase of pathogen or potentially pathogenic bacteria within that mucus layer. For us, when we get stressed, we come down with the flu. If a coral becomes stressed, they could come down with a number of different types of infectious diseases. Here's an example of some of the diseases that a Caribbean coral might fall prey to. These are the ones for which we do know a little bit about the causative agents. I have to say that we're back in the 19th century in terms of understanding the causative agents in terms of coral diseases. But here, this particular disease, white band disease in the Caribbean, was responsible for the loss of up to 95% of this dominant framework building staghorn or elkhorn coral in some locations, in particular the US Virgin Islands. And you can see here that along this front between the tissue and the exposed skeleton here, that the tissue literally peels off at rates of millimeters a day. In this next one, white plague, the white skeleton here, so this I'd like to emphasize is not bleaching. We're actually looking at the skeleton exposed as the tissue is stripped away. And in this case, the tissue is lost even more quickly at rates of up to centimeters a day. Interestingly, this one, white pox disease, has affected, again, the elkhorn corals in the Caribbean, and in particular around reefs in Florida, where the major sewage systems are septic tanks, which sit on the underground aquifers, and they drain into the local reefs. And this particular disease has been associated with a common fecal bacteria, which is able to also live in water as a free-living microbe in water and in soils. This one here, this particular disease, aspergillosis, is a fungal disease that has been decimating sea fan populations in the Caribbean for some decades and resulted in the loss of up to 3/4 of the populations in some reefs. And interestingly, again, there appears some of these fungal spores have been isolated in terrestrial runoff into the local reefs. So there are some emerging links, not all in all cases, but in some cases we're seeing some links between microbes on the land and then impinging into the local reefs and sea situations. Now, the classic epidemiological triangle has underlain disease ecology for many, many years, and this diagram encapsulates the importance of the environment in the interactions between three different players now in this diagram that shows the interactions leading to disease. So the host interacts with causative agents, and these can be either infectious or non-infectious, so we can have non-infectious agents. And the interaction is then played out on an environmental stage. And for a coral, the particular stressors in that environmental stage that we know about are increased temperatures, increased nutrients and sediments in particular. There are others, but those are probably some of the main stressors that are affecting or facing corals today. Where these three triangles intersect, that's obviously where the coral is going to have the greatest decrease in resistance to disease, and this is also where pathogens are likely to have increased virulence. The environment is important enough that it's actually nice to visualize it as a teeter-totter. So if we think of the environment as a fulcrum and the host and the agent as being balanced on that fulcrum, then if there are factors that that upset that balance between, and particularly environmental factors, that upset the balance between the host and the agent, then the agent may be favored, and as I said, this is where the host becomes more susceptible to disease. And that appears to be what happened here. Following a major bleaching event in the Caribbean in 2005, this is a once-in-a-century thermal anomaly that led to bleaching, widespread bleaching. Here in this particular study, in September, these corals were bleached. And I'd like to say that, again, the tissue is still present here, so that the coral, in this case, did recover. So by October, they had regained the zooxanthellae, those algae that live with the coral tissues. They had regained their normal, healthy colors. But then, a month later, they became susceptible to white plague, one of these infectious diseases, and within a month it had spread throughout this particular quadrat. So this is just a nice way of visualizing the same colony. This little arrow is pointing to the same colony in each of these quadrats here. Now by this time, the corals had actually lost the tissue, so this is not bleaching, they've actually lost the tissue, and ultimately then you see very rapidly algae and other sorts of colonizing organisms coming in, meaning that there's very little chance that these sorts of corals will recover in the near future. All right, I've been talking largely about diseases in the Caribbean. What about the Indo-Pacific and the Great Barrier Reef? This is a map showing the distribution of diseases in 2004 in the Global Coral Disease Database. And I think what you can see here is that there's a preponderance of red stars. So these red stars indicate disease records. The orange are the reefs and their locations throughout the world. But it's quite clear that the Caribbean at this stage looked to be a coral disease hotspot. It's certainly where the first coral disease was recorded. It's where the first epizootics were recorded. And it's where 66% of all the records of diseases were recorded, despite the fact that it only contains 8% of the world's reefs. And that's where, as I said, we came in with a program within the Center of Excellence to have a look at the state of play of corals on the Great Barrier Reef. And we've been undertaking an ecological program now on 3 different sectors throughout the Great Barrier Reef, from the northern sector in the north right through to the southern sector down here, working from some of these island research stations to try and have a bit of give the Great Barrier Reef a little bit of a medical examination. This is obviously a very daunting task when we're looking at something that spans 1,500 kilometers. So we've attempted to spread our effort across these 3 sectors, and also at reefs going from the coast out to the outer edge here, and this continental shelf varies between 100 and a couple hundred kilometers. Just to help you visualize what we do, we lay transect tapes which are 20 meters long, and then we go along those tapes on both sides with laying or delimiting a belt that's 1 meter on either side of that tape, and have a look at every coral within that belt. And so far, we've probably examined over 350,000 corals in the course of this 4-year study across these 3 sectors, 3 shelf positions, and at a variety of reefs and sites. And just to give you a bit of a mugshot of this gallery of diseases that we have identified in the course of these ecological studies, we've seen a number of growth anomalies that vary from these tumors sitting on these branching species to very bizarre-looking cases of growth that looks to have gone fairly out of control. We've seen ciliate diseases. So ciliates are one-celled organisms that, again, invade the tissues and advance in a front up here from, this is the mass of ciliates advancing up into the live coral tissue area. Another type of disease, in this case caused by a cyanobacteria that gathers as a front between the healthy tissue and the skeleton, and you can see it in multiple lesions on this coral. This is White syndrome is a name that's been given to a suite of, or a potential suite of diseases that may be caused by a range of factors, and including bacterial pathogens, environmental stressors, and cell suicide, something I'll say a little bit more about in a minute. Another disease caused by a different type of ciliate. In this case, you can see it swarming like masses of maggots and moving up, exposing the white skeleton. Here's the healthy tissue and the mass of ciliates. And finally, one that's been called Black Death, that starts out as a bleach spot but ends up with a very black mat of microbial communities. And in these surveys, what they have revealed is that diseases are widespread on the Great Barrier Reef. This just shows you that if we lump all the diseases together, then 100% of all the reefs that we've looked at do have some sort of disease. But in fact, 6 of the diseases do occur on more than 60% of reefs. So it does look like disease is widespread, but I want to make it clear that it's, in general, at very low abundance, low prevalence. So if we put together the results from those surveys in the northern sector here, and this is the central sector, going across from the inner shelf to the outer shelf over 3 years, the overall prevalence of disease is quite low, ranging from 2 to 3 degrees here in the central section to more like 3, possibly maximum 5%. So in general, and this one's for the managers, I would say that the Great Barrier Reef gets a bit of a tick on its medical exam. It would be surprising if there wasn't any disease. It's overall low abundance. But I do want to say that there are some causes for concern. We do see that diseases in particular are affecting certain families of corals. These 2 families are the ones that are the fast-growing branching sorts of species, and they're the ones that dominate our mid and outer shelf reefs. And so these are examples of these 2 families of corals that are particularly susceptible. And more than that, these are also the families that are particularly susceptible to bleaching and also to crown-of-thorns starfish predation. So they're ones that we can consider particularly vulnerable to changes involved with increasing temperatures and potential diseases. We do see also that there can be very dramatic impacts on localized scales of diseases. So this is an example of brown band disease, which is caused by a ciliate. And you can see the skeleton exposed, the band of ciliates, and the healthy tissue of the coral above. And through time, we see that within 48 and 72 hours that the coral is stripped, the tissue is stripped off the skeleton, and now a front that was down here is up almost to the branching point and then beyond the branching point. So it's progressing at rates of 2 to 10 centimeters per day up some of these corals, and then can spread throughout these patches of staghorn corals. Now, to understand these diseases, a powerful approach that has been made possible because there are people within the center working at many different levels, from that ecological macroscopic scale down to physiological changes and also cellular level indicators of diseases and microbial studies. We're beginning to understand that there are different ways in which diseases are affecting corals. And here's a bit of a summary. We see these first 3 diseases here. There do seem to be bacteria that are infiltrating the tissues when you look at a cellular level. And the tissues in these first 2 cases, at least, are affected by a necrosis, which means that there's probably a potential pathogenesis going on here. We're looking at bacterial pathogens, and probably as a result of an external attack, which is leading to the cell membranes necrosing. That looks very different from this last case, where there's no bacteria associated with this last disease here. There is with this one, but as well, when you look at the cellular level with these last 2 diseases, there is evidence of a host-controlled process, and in particular, rather than that necrotic tissue at the cellular level, we're now seeing the fact that the cells are being condensed, that there's evidence of resorption of the cellular contents, and what we call programmed cell death. And that's what we mean by cell suicide, that it looks like the host, potentially in response to a variety of factors. It could be environmental stress, it could be a pathogen that's causing the tissues to basically withdraw from that front, from that potential attack, and recede. The interesting thing with this particular disease here is that it doesn't seem to stop. It just seems to keep going till you lose the whole coral. Now, yes, corals do get sick, but perhaps the even more interesting question is, how do they fight back? And I think this is an exciting area that we're starting to work on within the center, that there are a number of different responses that corals can have in terms of their innate immunity, their ability to resist diseases. And one is the potential for inflammation response in response to a whole variety of stressors that do things like upregulate pathways that lead to melanin deposition. That's been shown to be important in that CFAN disease system that I showed you a minute ago, that you do get melanin being produced around these lesions. In this case, it looks like the melanin is walling off fungal hyphae that are invading through the skeleton and trying to get into the tissues. So the melanin actually walls off the pathogen. As well, you get amoebocytes that try and attack and contain the fungal hyphae. And lastly, I'd just like to say that also within our program, there's the work that Lena's already referred to of David Miller and his group, that there is evidence of other pathways in the innate immune system, things like toll-like receptors that we're starting to see. Now, going a little bit further afield, I just wanted to show you briefly that we are seeing disease outbreaks in other reefs throughout the Indo-Pacific, and in particular, I'll look at the Marshall Islands and Palau here. And as you fly into Palau, you are confronted with a very large atoll surrounding a lagoon. We were called by a colleague that was distressed to find he lived about here. This was his backyard here, which looked like this. And he would dive on this reef on a weekly basis. And he was distressed to see that a lot of these big plate corals were dying back. And in fact, when we went, we saw numbers of colonies 2 and 3 meters across that had this white syndrome, this advancing line, healthy tissue, white exposed skeleton. And he had tagged a number of these which showed the loss of tissue, the front starting here, going around the colony, so that in 6 months there was nothing left of this particular one. In this example, within 2.5 months, the initial lesion had spread until it starts to move quite rapidly. Here's the remaining bit of live tissue within the 2.5 months. All up, he tracked some 850 colonies, and the red indicates the ones that died in 2008. That represented more than 15% of the colonies, and about 16% of the coral cover that was lost. The blue indicates corals that still had active infections at the end of that year. There are lots of cases where it looks like it's spreading from one colony to the next, which would be a bit of a hallmark of an infectious disease. Equally in Palau, what's thought to be a fairly idyllic place, when we were looking at the baseline levels of disease, we happened across an outbreak again of a different type of a white syndrome. In this case, evidence of a very rapid loss of tissue. This is not bleach, this is actually exposed skeleton, and also affecting quite a range of coral species, and similarly emanating out from a point source. And moving even more rapidly, in the course of 1 week, you can see this plate that was alive here, except the disease front down here, completely dead. And here you can see that it had started to spread from this plate and continued on here. So basically, hallmarks of an infectious syndrome spreading from coral to coral. Just going back to the Great Barrier Reef briefly, there are evidences also of outbreaks that we're starting to see here. Some of these, this black band disease outbreak, you can see the black band over the course of 7 weeks in winter, going from a lesion this size to one here. The number of disease cases seems to be highest in summer, decreases in winter, back up higher in summer. Actually, we've gone past one here. Highest in summer, down to winter, back up in summer. That very clearly follows the temperature cycle, and not only in the number of cases, but here, the actual rate of progression. So highest in summer, lowest in winter. So there are correlations emerging between the rate of progression of disease and the spread throughout a population. As well, this information from the data set from the Ames Long-Term Monitoring Program, which is one of the largest programs monitoring 48 reefs up and down the Great Barrier Reef annually, showed an increase of something like 20% or 20-fold in the number of cases of white syndrome in 2002-03. This also correlated to the, in fact, probably the most severe bleaching event on the Great Barrier Reef. This shows the disease abundance in that 2002-2003 year, the highest abundance of disease on these outer shelf reefs. This shows the sea surface anomaly, temperature anomaly from satellite data. To make a long story very short, if we look at the number of thermal anomalies and the coral cover and the disease cases, you can see that there's a relationship. As coral cover increases and thermal anomalies increase, then we get an increase in coral disease. So there are emerging links between thermal anomalies, and in particular when it's combined with host density, another sort of hallmark of an infectious disease in this case. So just to summarize very briefly, going back to this diagram of the relationship between the host and the agent being balanced on a fulcrum, which is the environment, what we're seeing potentially is a series of threats to the coral host, which is aligning with the agents, so warming oceans, acidification of seawater, storm damage as we see increased, or what are predicted to be increased intensities and frequencies of cyclonic storm activity. And then if we add to that poor water quality, then you can see that we have the potential to tip the balance to sorry, it's sticking here. Tip the balance so that the agent becomes favored, the host, the resistance goes down, and we may see more cases of diseases affecting corals. Which leads me to the final conclusion that, yes, we are seeing that coral diseases are widespread on the Great Barrier Reef, but I would like to make the point that the overall prevalence is low, that we are seeing disease outbreaks here on the Great Barrier Reef as well as elsewhere throughout the Indo-Pacific, but on fairly localized scales at present. But things like water quality, ocean warming, and other predicted climate impacts are likely to increase the prevalence and severity of coral disease. So we need to do things like support initiatives like the Water Quality Management Program, plan to keep the water quality at a level that's not going to cause additional stress, and of course to address issues that might help to bolster the health of corals in the light of climate change. And finally, acknowledgements. All right, thank you.
Time for one question.